Okay, we should start. It is a great pleasure to begin these online presentations of the Josep Carreras Leukemia Research Institute. Times have changed. We'll have loved to have José Luis here in our place in a physical manner, not in this virtual manner, but this is how it is live right now. José Luis Gómez-Skarmeta is full professor at the Spanish National Research Council, the CSIC, at the Andalusian Centre for Developmental Biology in Sevilla. He has been working for more than 29 years in biology. He has published many important papers, and he's really at the forefront of the developmental biology and evolution, functional genomics and epigenomics field. He's one of these rare avis or mavericks that is able to combine evolution and epigenomics. That is something extremely difficult, I would say. He has received several important recognitions for his work, among these the Garcia Advanced Grant and the Carmen and Severo Ochoa Molecular Biology Award. So it's a great pleasure. He's also an EMBO member. He's also the scientific director of the Maria de Maeztu Unit of Excellence and the current director of the Department of Gene Regulation at his institute. So it's a real pleasure to introduce Dr. José Luis Gómez-Skarmeta. José Luis, all yours.
Thank you very much for the invitation. It's a real pleasure to be here in this online seminar with such a big audience. It's very nice. I'm sorry that we cannot meet in person, but you know, the different circumstances are how they are. And today, despite your institute is focused on cancer, our work is basically evolutionary. I will try to give you a different perspective from the epigenomic view of how epigenomics can lead to understand how evolution have progressed and have contributed to body plan changes. So the critical question we want to ask in our lab is how different body shapes are generated during evolution. And you know that basically all vertebrates are constructed by the same toolkit of genes. So at the end, the final morphology is a result of how you use these genes during development, how you activate and repress those genes during the construction of the body plan. So that's why we need to address this from the epigenomic point of view. So we started this type of work a few years ago, and we call developmental epigenomics. And we basically start by doing mapping some epigenomic marks associated to promoters. Let me check here. Let me change this to this. Epigenetic marks associated with promoters or active enhancers at different developmental stages during several phase development. The idea was to try to look for the dynamic of gene regulation during development in a model organisms. And I think that this snapshot represents very well what we found in these experiments. So here we are looking at the region around a developmental gene, SOX2. It's a gene that is required for multiple processes during development. We are looking at different developmental stages here, which is blastula, gastrula, organogenesis, and later organogenesis. And this is epigenomic mark associated to promoters. And you see the gene is active all through development. And this is a K27 acetylation, which is a mark associated to active enhancer. And you can see that at earlier stages, so the gene is active all through development, but at earlier stages, there are some enhancers that are being active and then are shut down. And they are probably required for the early expression of the genes everywhere and to keep this gene involved in the process of pluripotency. But you cannot keep pluripotency forever, so you have to change the expression pattern of the genes. So you need to shut down some enhancer and activate new ones, and this will be responsible for the late expression domain of genes. So the point here is that I want you to take from this snapshot is that the regulation is extremely dynamic, and this regulation, especially around genes that are constructing the body plan, which we call developmental genes, which are a transcription factor or signaling molecules, this regulation, the dynamic of this regulation will change the dynamic of gene expression that we will trigger the process of development. So development itself is the final endpoint of the dynamic of all these the dynamic cis regulatory elements. So looking at the dynamic of these regulatory elements and compare them in different organisms, we'll be able to understand evolution, because evolution is not other thing that changes of gene expression during development. So in order to change body plan formation or morphologies, you need to change gene expression during development. So this is why we are so interested in the dynamic of gene regulation during development and in a comparative way in different organisms. So in addition to know where the regulatory information is present, we also need to know which are the genes that are being affected by this regulatory, because here I can tell you that these enhancers are likely controlling SOX2, but they may be well also controlling nearby genes. So for that, there are new technologies which are like based on chromatin conformation capture, and this is one of them. This is the 4C map. We're looking at the same gene, SOX2, zoom out. We are zooming out the genome. So this is promoter mark, this is enhancer mark, and here are a map contact based on 4C-seq. 4C-seq allows you to map all contact made by one particular region of the chromosome, and in this particular case, is the SOX promoter. So we are looking at where are the SOX2 promoter contacting with in the genome. And you see that the region is limited in the space. So not all genome is in contact with SOX2. There are a particular region of the genome, which is quite large here, it's about 500 kb. And there are regions that have more contact and less contact. And this is what we call the regulatory landscape, the territory of host SOX2 that may contain all the enhancers that are controlling the expression of the genes. So the genes, all the genes in vertebrates, and in fact in most metazoans, are compartmentalized in domains which are called topological associated domains, which are these regions in which chromatin contact are favored, and these contacts allow the distal enhancers like this one finally come in proximity with the promoter, and they can really reach the promoter. So in order to understand evolution and evolution of body plan, we need to understand how different regulatory elements may change during evolution, but also how the regulatory landscape itself can change, because during evolution there are a lot of recombination, translocation, inversion that may remove large pieces of this regulatory landscape, or may bring together territory that is compatible, that will be a source of new regulatory elements. So we will address this issue during this talk, and for that we will concentrate basically in this transition between invertebrates to vertebrates. Okay, this is an Amphioxus. Amphioxus is a cephalochordate. It's a living organisms, invertebrate living organisms, which is likely most close to the ancestral of all vertebrates. This transition is very important because many features, characteristics of vertebrates were developed in this transition, like face structure, limbs, and several organs, etc. And it go together with a very important genomic event, which is a double duplication of the genome. So in this transition, the genome was duplicated twice. So many genes were kept after the duplication as 1 copy, 2 copies, 3 copies, or 4 copies. And genes that were maintained as multiple copies are mostly developmental genes. So genes required for constructing body plan have gained in number because after this duplication. At the end, the number of genes, the final number of genes is not as similar between invertebrate and vertebrate, but the number of developmental genes have likely increased quite a lot in vertebrates. So we want to know how these genes have changed in terms of regulation and expression, and how these changes may have contributed to the generation of vertebrate-specific morphologies. So for that, as I told you, we started a few years ago doing a whole map of epigenomic marks in zebrafish at different developmental stages. We have also RNA-seq from different studies. We have methylomes, polycomb marks, attack signal, which is a readout of open chromatin, and we will use it here as a readout of regulatory information. So for us, attack, open chromatin will be considered as regulatory elements or active regulatory elements. So we have all this data that we have produced during the last years in zebrafish. So we want to do the same in Amphioxus. So for that, we sequence the European and Amphioxus genome, and we produce quite a lot of RNA-seq data at different stages and different adult organs, and also epigenomic information at different developmental stages. So the idea is at the end up with a set of data, similar set of data in both species that can be now compared and try to use this comparison to understand the process that took place in this transition and how this have influenced gene expression and probably body plan formation. So let me show you just an example of the data. This is a snapshot of the Amphioxus data. We are looking around the PAX9 gene. The PAX19 is the ancestral gene for PAX1 and PAX9 invertebrates. And we are looking at different RNA-seq data, open chromatin, ataxia data, enhancer mark, promoter mark, methylome, etc., etc. And let's concentrate in these two regions, which are open chromatin, that are likely regulatory elements. We know that in general, these attack peaks do correspond to regulatory elements because we have tried quite a lot. And this is an example of difference. We have tried them in zebrafish and Amphioxus. These are zebrafish transgenics for different elements that we have selected. Forget about this expression in the midbrain, which is our positive control. Look at this. These two particular regulatory elements are these two guys that drive expression in the pharyngeal arc, which is a region in which this gene is in vertebrates. So this makes sense. But we have also some transgenic done in Amphioxus. These are Hox regulatory elements that are expressed in the nervous system. These are enhancers that are expressed in the muscle, both in zebrafish and in Amphioxus. And this is another one from FOXA that is expressed in endoderm, both in Amphioxus here, which is in the endoderm, and in zebrafish. So we know that this open chromatin region can be considered as potential enhancer, and we will concentrate on that. And one thing that we can remark here, if we count the number of attack peak that we found in zebrafish or in Amphioxus, we have quite more potential regulatory element in zebrafish. And they mainly locate in intergenic distal regulatory elements. So this can be done better if you just divide it by gene. So you count the number of regulatory elements per gene, and you can see that in zebrafish, you have more regulatory elements per gene than in Amphioxus. But this increase in the regulatory information is not the same for all genes. Remember, I told you in this transition, the genes, the genome was duplicated, and some genes were kept in vertebrate as multiple copies, 1, 2, 3, 4. And we have found that depending on the number of copies retained in vertebrates, you have more or less enhancers. So here we are looking at the number of regulatory elements associated in Amphioxus genes that have only one copy, but will give 1 copy in vertebrate, 2 copies in vertebrate, 3 copies in vertebrate, or 4 copies in vertebrate. I told you that most of these retained genes are developmental genes. So because they are developmental genes, they have more complex regulation, and they have already more enhancer in Amphioxus. But if you go to vertebrate, you see that they gain more regulatory information as more copies are maintained. So not all genes are gaining regulatory elements in the same way. Some genes are gaining more regulatory elements. And in fact, if we compare not for each gene the different copies, we will find that one copy or some copies acquire very little number of regulatory elements, or very similar to that compared in Amphioxus, while others gain a lot of regulatory information. So there are an equal distribution of this regulatory information. Usually, a few of the copies retain lower number of enhancers, and other copies start to gain regulatory information. Okay, so this is important because we will now try to integrate this with what happened with the expression pattern of these genes. And for that, we will basically concentrate in a group of genes that in Amphioxus are expressed in 9 adult tissue. And if we take all the genes all the copies that derive from these ancestral genes in vertebrates, recapitulate also the expression in all the different tissues. But any genes, genes in particular, the different genes may have all or different expression patterns. So altogether, recapitulate the ancestral expression, but it could be that both genes or several genes contain all the expression patterns. Different genes of this duplicate are so functional. One retain one expression domains and the other one retain other expression domains. And it could be also the possibility that one copy retain the ancestral expression and the other copy become specialized. So become very restricted expression pattern. So we can count, because we have about a family of 300 genes of this category. We can really count the number of events that are redundant, functional, and specialized. And we found that in general, no matter if you compare Amphioxus expression with expression in mouse, frog, or zebrafish, in most cases, genes become specialized. So you have that after the genome duplication, one of the copies retain expression in all ancestral expression domains, but the other copies become much more specialized, become much more restricted in expression pattern. And if we look now, what happened with the protein, the coding protein of these different copies, so we can see that here we are comparing the sequence of the coding region with the ancestral Amphioxus sequence. So those genes that are redundant have very little change in terms in the coding sequence compared with ancestral. So it's very close to 100% similarity. Those genes become subfunctional, change much more. There are much more variation in the coding sequence in these genes that become subfunctional. And if we take this set of genes, we can separate the gene that retain the ancestral expression, so the one that is more similar to the redundant gene, so the one that is more similar to the ancestral expression, and this change much less in terms of coding sequence than the genes that become more specialized. And the more specialized you become, the more restricted is your expression pattern, the more variation you find in the coding sequence. So this is telling you that after genome duplication, you have a backup that retain the ancestral function, and the other genes that start to accumulate new regulatory information and also become specialized in terms of expression pattern and become much more divergent in terms of coding. So you have other elements to play around because you have a backup copy that retain the ancestral function, which is this one. Okay. So where is this specialization taking place preferentially? Is found basically at the brain tissue. So brain is a region that is enriched in genes that become very much specialized. And we can also count now the number of enhancers associated to these different genes. And we found that the number of enhancers associated to the genes that have redundant expression is low compared with the number of enhancers of the genes that have been subfunctionalized. And then if we go to this collection of genes, the copy that retain the ancestral expression have less enhancer, and the copies that become much more specialized gain more enhancers. So you need more enhancers to generate more specialized expression domains. And this is one example. I will show now 4 copies of that derive from ancestral gene in Amphioxus. There's one copy, this one, that is retain a generalized expression like the Amphioxus one, but you can see that the other copies become very much specialized in terms of expression pattern. So you have this like as a backup of the ancestral function, and with the other ones, you can really start to play around in terms of expression and function by accumulating variation in the coding sequence. And in the same in terms of regulation, if we look at the number of regulatory elements associated to these genes, here we are looking at this gene. Green are promoter mark, this epigenomic mark associated to promoter. These are enhancer mark, and this is open chromatin in black. And you see that this gene has a couple of enhancer, while this one that has a more complex expression in the neural tube has much more enhancer associated. So the take-home message of this part is that multiple novel cis regulatory elements have been incorporated to the vertebral lineage. Okay, and these new regulatory elements have been contributed to the specialization of duplicate genes. So different duplicate genes have gained different number of enhancers, and these enhancers have helped to restrict and make these expression domains more restricted. And this specialization of duplicate genes, we believe that are a critical issue that have contributed to generate new vertebrate-specific cell types and tissues. Okay, so from this part, and it's important to take the take-home message, this transition that is associated with duplication of enhancer is also associated with a gain of regulatory information that have been used differently in different copies to make genes much more restricted and make them be contribute to probably to new tissues. But we want to know now which type of regulatory information is the one that is coming up. Which are the new enhancer? What are these new enhancer coming from? So what type of regulatory information is gained in vertebrate? And we center in signaling pathway. Why in signaling pathway? Because the signaling pathway is a really limited number of pathways that are evolutionarily conserved in all metasomes and are controlling many different gene regulatory networks. So they are at the top of the hierarchy of many gene regulatory networks, despite there are a few of them. So they are involved in multiple processes. So we try to compare how this signaling pathway may have evolved in this transition from invertebrate to vertebrate. Okay, and for that, this is the experimental approach we have done. Basically, we have treated zebrafish or Amphioxus embryos during gastrulation, from early blastula to late gastrula, with different drugs that either stimulate or interfere with signaling pathway. We have used retinoic acid to stimulate a retinoic acid signaling pathway. We have used bio, which is an agonist of Wnt, so this gain of Wnt signaling. We have used this one, which inhibit FGF signaling, and this one, which inhibit nodal signaling. So we are interfering with all the signaling, the same drugs in both animals. And after gastrula, we have done ATAC-seq and RNA-seq to find the genes that are responding to this perturbation and the regulatory information that change upon this perturbation. Okay, so first let's go to some controls and some important data. One thing that we find is critical is that no matter which signal pathway we are looking at, in all cases we find much more vertebrate genes with gene ontology which are affected by these drug treatments, which have a developmental or signaling pathway gene ontologies. So we are affecting, you can see here, the number, we have divided the genes into metabolic genes associated to signaling pathway or associated to development pathway. And you can see that in vertebrate, in zebrafish, in all cases, we have much more of the signaling pathway at development terms. So we have more developmental genes and patterning genes responding to this signaling pathway in vertebrate than in invertebrate in all these treatments. But let's see first if these treatments are trustful, if they are really affecting the genes that they should affect. So for that, we're using this type of graph. Basically, we are taking all the gene ontologies associated to these genes, and this graph group this in gene ontology by proximity in a 2D chart, and here group together those that are close related. Here they are the development. We are just marking the development gene ontologies. So you see that there are many developmental genes affected by these drugs, and these are examples of the genes, some of the genes and some of the processes that are affected by this drug. And this process made totally sense with the signaling pathway. For example, retinoic acid is affecting the retinoic acid signaling pathway. Nodal, which is essential to endodermis and mesodermal formation, is affecting pancreas and mesenchymal formation. FGF, which is critical for muscle formation, is affecting muscle formation. This is an Amphioxus. Or Wnt is critical for many things and then in neural development, and you can see many genes, developmental genes involved in this process that are affected by this treatment. Now, what happens when we do the same experiment, or we look at the same experiment in zebrafish? We see that the number of terms and the number of gene ontologies associated with development have largely increased. So we have much more gene ontology terms associated with development, much more number of developmental genes that are responding to this signaling, and the gene ontologies of these genes affected also make sense with the affected pathway. Okay, for example, retinoic acid is critical for hindbrain development, otic formation, etc., 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 etc. So this is just a control to show you that the treatment are making sense and are affecting the right genes in both systems. So we can really do comparative analysis. But we have to also look at the attack peaks, so at the regulatory elements. Other regulatory elements affected by this treatment making sense, and for that, basically here we are looking at the motive and reach in those regulatory elements that are affected by this treatment. So when you perturb Wnt signaling, when you increase Wnt signaling, you find TCF in both invertebrate and vertebrate, one of the top motif affected. And together with that, other genes that are known to be downregulated are very early in the pathway controlled by Wnt in this process. Retinoic acid, the same. You find retinoic acid receptor, one of the most important motif, and Hox genes, which is a direct target of retinoic acid, etc., etc., etc. So we know that the treatment are working well in terms of the genes affected and the regulatory element that are affected. And now we can put everything together. So the idea now, let's find the collection of genes that are perturbed by each individual signal that also contain an attack peak, so a regulatory element that is also affected by this signaling pathway. So these are the core genes that are reading the response to this treatment in a better way, in a more clear way. And then we take these genes, we get the gene ontology associated with the gene, and we cluster them. The idea is to find to what extent the different geoterm different process require 1, 2, 3 or multiple signaling pathway. And this is what you get in Amphioxus. In Amphioxus, these are the terms we can recover, different developmental terms. There are some developmental terms, but there are many others that are not development by itself. And you can see this degree, the intensity, the value, the contribution of the signaling pathway to the process. You can see that, for example, retinoic acid signaling pathway is enriched after treatment with retinoic acid, and in general, you see that there are different processes that are affected by 1, 2, or even 3 signaling pathway. But there are not that many processes here in Amphioxus, especially when you compare the same in zebrafish. If you go to zebrafish now, you see that you find much more geoterms because you have much more genes affected, and you have much more interrelation between signaling pathways. There are processes that require all different pathways. There are much more interconnection between different pathways. So this is revealing, or this is pointing at a more integration of signaling pathway, more interconnection of signaling pathway in vertebrate than in invertebrate. And we can count this very simple. So let's take only the set of RNA-seq data. So let's look at the number of genes that are affected by this treatment at the RNA-seq level by 1 signaling pathway, 2 signaling pathway, 3 signaling pathway, or 4 signaling pathway. In Amphioxus and zebrafish. Amphioxus here is in green, the vertebrate is in blue. And you can see that the number of genes affected at the RNA-seq level by 1 signaling pathway is higher in Amphioxus than in vertebrate, but the number of genes affected by multiple signaling pathway is higher in vertebrate than in Amphioxus. So this is just taking the RNA-seq level. Now, if we combine and we select the genes that contain an attack peak also associated to the gene that is affected, so genes that have a regulatory element and also respond at the RNA-seq level, and we do the same, we find exactly the same process. So basically, the invertebrates, which is the blue charts here, invertebrate, you get more genes that are responding to more signaling pathway. And this is much more clear view when you just draw the interaction between signaling pathway. Here, we are looking here in Amphioxus and in zebrafish, the number of genes that are regulated by Wnt, the number of genes that are regulated by nodal, FGF, of retinoic acid, the genes are regulated by retinoic acid and Wnt and FGF, etc., etc., etc. If you go to vertebrate, you see that these nodes are much more enriched. You have much more genes that are responding to multiple signaling pathway. There are much more interconnection of different signaling pathway in vertebrate than in invertebrate. And in fact, we can also try to ask which are or where are, is there any relationship with this interconnection degree with the expression domains where these genes are found? So we have moved to single cell data in zebrafish, and we have looked at the degree of interconnection in tissues that have a more ancestral origin, like muscle, liver, mesoderm, intestine, and you can see that there's not that much difference between the genes that have more connections or less interconnection. So basically, they are basically the same. Genes that are not special enrichment in this tissue for genes that are that have multiple interconnection between signaling pathways. But if you go to the novel tissues that are very specific to vertebrate, now you start to see that genes that have multiple interconnection between signaling pathways tend to be enriched in those tissues more than those that have a single connection. So this interconnection of signaling pathway, which is a consequence of gaining new regulatory elements in the transition between vertebrate and vertebrate, have allowed this gene to generate more complex expression patterns and also to be expressed in tissues that will generate new territories, new organs in vertebrates. So the conclusion from this second part is that the incorporation of these novel cis regulatory elements in the vertebrate lineage to the regulatory landscape of many developmental genes have allowed these genes to now respond to multiple signaling pathways in the vertebrate lineage. So instead of responding to one signaling pathway, many of these genes now respond to multiple signaling pathways. By integrating multiple signaling pathways, they now can generate more interconnectivity and probably contribute this to the generation of more tissue-specific cell expression that is associated to the generation of vertebrate-related cell types in the vertebrate lineages. So this gain of regulatory information is basically allowing interconnection and gain of complexity by mixing the activity of different signaling pathways and generating very restricted expression patterns that help to generate new tissues and new organs. So at the end, the increased signaling pathway interconnection in vertebrates likely facilitate the restriction of the expression domains of developmental genes. And this in turn is expected to contribute to the increment of the tissue complexity required to generate vertebrate morphology novelties. So we know now new regulatory elements come, so duplication of the genome, new genes, these duplicate genes gain regulatory elements in a different proportion. Some of them gain a lot of regulatory elements. These regulatory elements integrate signaling for multiple signaling pathway, and this helps the generation of very complex expression pattern. But how now can we relate this gain of regulatory information with the 3D architecture, with the regulatory landscape of the developmental genes? And for that, we start doing some High-C experiment at the beginning. Here, I'm showing you High-C data from Amphioxus and from vertebrate and from Amphioxus. You cannot see very well here, but this is a host cluster in Amphioxus. This is one host cluster in vertebrate. In vertebrate, the host cluster is in the middle of two topological associating domains, which are defined by this High-C structure. And in vertebrate, there are only one single tab, including the whole cluster. So in this transition between vertebrate and vertebrate, a new tab become integrated to the regulatory landscape of developmental genes like the host. So we like to try to understand this in a more general way. We started with the High-C, but we realized that the definition was not that good, so we moved into high-chip data. High-chip data is basically a High-C, but instead of sequence all the High-C library, so all the contact between all different regions of the genome, you do an intermediate step of purification with an antibody that can be, for example, K4 methylation. So you can enrich for the contact from promoter. You can enrich from the contact from enhancer or from polycommon regions. So you reduce the number of the library by looking only at the contact made by different epigenomic map. In this case, we will concentrate at the contact, the high chip data from K4 trimethylation, which allow us to look the genome-wide, in the genome-wide, the regulatory landscape, the contact made by all active promoters in Amphioxus and invertebrate. And now we can compare data between the two species, okay? And this is something that you basically get here. Here is the size of the regulatory landscape, of how long is the contact of the promoter with the nearby non-coding domain in Amphioxus and in zebrafish. And with this Amphioxus, these are zebrafish. We have divided the genome, the genes in two sets, housekeeping genes and developmental genes. And you can see that in invertebrate, already developmental genes has larger regulatory landscape because developmental genes contain more regulatory information than housekeeping. If you move to vertebrate, you see the same, and you see also that especially developmental genes gain in size with respect with Amphioxus. And again, we can look this, not only this is a general view, but we can look now at what happened at those genes that are kept as 1 copy, 2 copies, 3 copies, or several copies. And we find that the number of the size of the regulatory landscape is higher in Amphioxus, depending on the copy that will be kept in vertebrate. So these are genes that in Amphioxus have 1 copy that will have 5 copies in Amphioxus. And you see it has a larger regulatory landscape than the genes in Amphioxus that will generate only 1 copy after the duplication. So this is because these are basically developmental genes. But if we move now to what happened to the size of the zebrafish genes, you see that the trains is the same, but the vertebrate genes that are retained as multiple copies can have larger regulatory size. Okay, so it's the same that we were seeing with enhancer at some point, but now we are looking at the level of the content made by the promoter. And in fact, if we divide the different genes in vertebrate in the ones that are, these are Amphioxus genes, the Amphioxus original gene, and these are the different genes. These are the 2 genes generated in zebrafish, a gene that generates 3 genes in zebrafish, a gene that generates 4, and the gene that generates 5 genes in zebrafish. And you see that usually it's not that all genes gain or enlarge the regulatory landscape, but in fact there are a few that stay as small or even smaller than ancestral one, but there are 1 particular copies that accumulate or increase more the regulatory landscape, which makes sense. We were looking at the enhancer, and indeed now we can count the real enhancer in this regulatory landscape, because before we were assigning enhancer by proximity. Now we can count the real enhancer that are in this regulatory landscape. And we find that, of course, the larger are the regulatory landscape, the more enhancers, the more attack peak this gene has. Okay, so the critical point is that the gain of regulatory information in this transition, in particular copies of the duplicate genes, go in parallel with an expansion of the regulatory landscape of that particular genes, especially developmental genes. But we have to consider, and now close to finish, we have to consider which are the tissues in which we are finding this type of event, this gain of increase of regulatory landscape, and you see that they are basically, to a large extent, neural tissue again. So neural tissue is a tissue, is a organ, let's say, that gain a lot of regulatory information, and this regulatory information influence the expression of the genes and influence the regulatory landscape of many developmental genes in the vertebrate lineage. Okay, so this is important, it's nice. So adding new enhancers, increase the regulatory landscape, but what happened with changes, large changes in the regulatory landscape? So we can use synteny to study. Synteny is the distribution of 2 genes along the genome in different organisms. Here you have 1 gene, for example, that may contain multiple enhancers, that may be developmental genes, multiple enhancer that may colonize the intron of the nearby gene. If this happens during evolution, these 2 genes will be kept together all through evolution. Okay? So we can look at the synteny how synteny have changed between invertebrate and vertebrate, with the aim to try to find vertebrate-specific synteny pairs. So changes that are genes that are together only in the vertebrate lineage. So recent event of integration between two genomic domains, and also how this new effect, new syntenic pair, have affect the regulatory landscape of the genes that are affected. And I will show you a few cases because we have detected about 400 new rewiring process in the vertebrate lineage. One of them is the one I showed you before in the host cluster, in which a whole tab become incorporated to the host cluster in the transition between vertebrate and vertebrate. And I will show you a couple of examples now. One of them is this one, patch. Patch is a gene that is a developmental gene, is a regulator, is involved in the sonic hedgehog pathway, is a receptor of the sonic hedgehog pathway. This is the centennial region in vertebrate around the gene. And this gene that is nearby patch is present with patch only in the vertebrate lineage. If you go to invertebrate, patch will not be associated with this gene. So this gene came together to the patch locus in the transition between invertebrate and vertebrate. And here we are looking at the chip set data, high chip set data from patch. These are the contact, this diagonal are the contact made by the patch promoter in several. And you can see that it extends to this side and to this side. So this territory that is new in vertebrate is in contact, direct contact with the promoter, and may contain critical regulatory information for patch. So let's look at whether this is the case. And for that, let's concentrate one process, which is vertebrate specific. It's the formation of the limbs. The limbs are present in vertebrate, but not in invertebrate. So now what we are looking here is, again, these are mouse data. It's the patch locus, this is the patch, this is the gene that is close to the patch. And we are looking here to two tracks. This track is a TAG-seq, open chromatin from limb bands. So this is revealing potential limb band enhancers. Limbs are specific for vertebrates. So, and we are finding that there are quite a lot of limbs, and patches required and sonic hedgehog is required for information. And you see that there are quite a lot of enhancer, limb enhancer associated with around the patch locus. And we're also looking at the chipset of TD3, which is a controller of the signaling pathway. And you see also several peaks around in the area. And in fact, all these three lie in the territory which is only present in vertebrate. So we don't know which is the function of this regulatory element, how's the contribution to the expression of patch in the limb, but we know that the new territory that was incorporated in the vertebrate lineage has been a source of enhancers that are required to control the expression of this gene in a tissue that is specific for the vertebrate. So this translocation, this incorporation of new regulatory material may have contributed to the function of patch in the formation of this particular vertebrate-specific structure in vertebrates. We have also looked at where are these rewired genes expressed using single cell, and we found that they are expressed basically enriched for neural tissue, but we find several vertebrate-specific territories that are enriched for these new rewired genes. So this new rewiring, these new translocations may have contributed to the expression of these genes in these tissues that are vertebrate-specific. And I will show you one last example, and I will finish with that. This is the COL11A gene. We are looking here at promoter marks, and this is a high chip for the promoter. These are the contact made by the promoter. This diagonal reflects the territory that is in contact with the promoter of this gene. This nearby gene is specific there in the vertebrate lineage. So this territory is newly associated to the gene only in the vertebrate lineage. Okay. where is expressed this gene? This gene is expressed in the neural crest, which is a very specific territory. This the single cell data. You can see that expression is here, and you can see it in situ how the gene is expressed there. So, is this new territory that has been incorporated in vertebrate a source of potential regulatory elements that are controlling the expression or are contributing to the expression of this gene in the neural crest or not? We can use some epigenomic data that is not from our lab. It comes from this paper. In this paper, they did in zebrafish attack on SOX10 cells, which are neural crest cells, and we are comparing this potential enhancer with whole embryos to the different stages. And you can see that there are several neural crest enhancers around this gene. So this gene is controlled by multiple neural crest enhancers, and these particular enhancers are in the region that is vertebral novel, in terms that have been landed there in the vertebral lineage. So we believe that this type of rewiring is bringing a compatible or a source of a non-coding DNA, which serves as a substrate for enhancers, new enhancers that probably integrate multiple signaling pathway and are contributing to new expression domain of developmental genes or non-developmental genes in vertebrate-specific tissues. So the final conclusions are that in the vertebrate-to-vertebrate transitions, genes gain regulatory input, some from, oh, I don't see, some from them come from signaling pathway. And this is associated with more restricted expression domains, which is also causing the expansion of the regulatory landscape of the genes that incorporate these new elements. And many genes are also rewired in the vertebrate lineage, incorporating new regulatory information that may have contributed to expression to vertebrate-specific tissues. So finally, I would like to thank the people that have contributed to this work, my team plus other team, because we are two together. This is the people that have contributed to part of this work. Nacho has now his own lab, and Rafa is doing his product, but all of them have been essential for most of the work, and these have been our collaborators, which have been critical for the Amphioxus project. Ferdinand assembled the genome, Hector provided Amphioxus embryos, Manu was critical for all the epigenomic analysis. Osren helped us with all with Ryan methylome analysis, Boris with promoter analysis, and Peter Holm with the design of the paper. And now I will thank all of you, and I will be more than happy to take questions. So send me the question to the chat, and I will be answering them one by one. I am alive or not?
Yes. Luis, thank you very much. It has been a great lecture, and I think that you can. There's a way to make questions that you have here in the system a question mark that you can write your questions for all the attendees. In the meantime, while they're preparing these questions that are almost there, I have just one general question so it's a very general question, really. I think this is great work. The difference between vertebrates and non-vertebrates, it is clear. They are probably more regulated at this level. And I have this question, Olga, about when you show your data. When you show your data at 4CM, for example, can you hear me?
No, yes.
Yes. Okay. So a lot of emphasis has been always done for the enhancers, and you mentioned a lot of the enhancers. Okay. And so what happened with the repressors? There is very little known, because all the time yourself and myself and many others will talk about enhancers, but we disconnect about the repressors at distance. Okay, and what is this? There are less, or this is a question of the field? How it works?
I think that the repressor itself, they themselves are not individual entities. So enhancer is basically a region of the genome that integrates many data, positive and negative. So the same regulatory information can be used as a repressor or as an enhancer, depending of the degree of factors that are binding to. So there are transition between the activity, but you cannot really, it's hard to find. And in fact, there are a couple of papers that show this year, I guess, that show that many of these regions that were detected as potential repressor were indeed enhancer in other tissues. So I think that you cannot really distinguish between repressor and enhancer. It's basically a regulatory element that integrate data in a positive or negative way.
Okay, and how it fits in all these, the insulators, how it fits?
Well, insulators are also, I don't think that insulators are also particular entities. I think that insulators are domains in the chromatin that are required for compartmentalization of the chromatin that can be act basically dividing the chromatin in these territories, these regulatory domains, and by helping this division, they act as insulators.
Okay.
This is a general view. This is a general view. Of course, you have to go to other layers there because you can go into methylation process that can do really active repression there. So there are sequences that with the repression, with polycom, and this really act on they do actual repression on that. So you have to be careful about that. But I think everything is somehow related with organization of the chromatin and activation or modulation of the regulatory activity of cis-chromatin automotors.
Okay, so I'm going to read the questions for you because I think I have access to the questions here. Okay.
Yes, because I do.
Yes, because you don't have access.
No, I don't have access to this.
Yes. Yes. There's a particular question about all this more complexity in the vertebrates. If this also relates to the immune system and genes involved in immunology.
This is something that we have not really focused on. Of course, I have concentrated mainly in mentioning developmental genes because developmental genes are involved in body plan formation. But definitely, developmental genes are not the only genes that are gaining this complexity in terms of regulation. In fact, many of them are not developmental genes, and I'm sure that immunity is also part of the process. So I think all the advance of the novelties, the functional novelties that you can find in tissue organs or system in vertebrates are suffering or are being benefited by this gain of regulatory information, which at the end, which allows basically generate more complex structure, more complex input from different signaling pathway and different environment conditions.
Yeah. There's another question that it says, here it is. Oh, go up here. Here it is. Here. No. Here. It says what it means that the metabolic genes in vertebrates are less affected by the disruption of the signaling pathways.
I mean, I don't know. I just no. This is not true. This is not true because in Amphioxus, in invertebrates, in fact, we find that the response to the perturbation of signaling pathway is highly enriched for metabolic genes. But this could be also due to the type of organism. This is an organism that is that has no skin, so it's very exposed to the environment. So the drugs come in and affect many processes at the same time. So I would not say that this is specific to the developmental genes, affect other processes, but in general, the point here is that, and what I want to remark is that in order to generate patterning, complex patterning, you need to gain more regulatory information, you need to gain more restricted domain. Of course, everything is integrated because it's a system. And metabolisms need to be integrated, and proliferation need to be integrated with patterning at some point. So other genes involved in other processes are also affected by the process. But in general, what we really see is that they respond to this signaling pathway during a developmental process, because remember, we are doing the experiment in a developmental window, are mostly developmental genes, especially in vertebrates.
Yeah. And for people working here in cancer, the way you compare vertebrates, non-vertebrates about this rewiring, so can this be applied in the same comparing, imagine a B cell and a B cell leukemia? And what is the right way to do it?
Oh, this the way. The way is to try to do maps of interaction like HiC, or I mean, it's well known now how translocations that fuse or divide topological associated domains, which basically what we are looking using high chip instead of HiC, but how these processes are clearly involved in many pathologies, including cancers. So obviously, we have, all of us, a lot of breakpoints if we compare one to us. Most of them, fortunately, take place at the TAT boundaries, so they are not affecting gene expression. But those that fuse borders or generate new border or expand the regulatory landscape of any genes are very, very much dangerous. And MYC is an example of how there are fusions or translocation that are providing new cancer to MYC that can now activate elsewhere, and this is helping or is contributing to the cancer progress. So obviously, human diseases are very much affected by this type of rewiring of the chromatin by changing regulatory landscapes.
And there's another question: there is a direct correlation between the number of cell types of an organism and the complexity that you are seeing here? Because now there are all this. There's a new paper showing that we have 850 cell types, or all this. So this is a direct relation, and the difference between vertebrates and non-vertebrates relates to the number of cell types.
I do believe so. I mean, for this lacking, it's lacking. There are some data from other invertebrate species, is that the complexity in terms of gene expression, which at the end is what it will define in a single cell experiment, the different cluster. The complexity of gene expression in invertebrate is less than in vertebrate. So you will increase the cell type. What is a cell type? A cell type at the end is the combination of different genes expressed together at the same time. Obviously, the more genes you have to express in different way, the more complex will be tissues. And this happens with tissues that are specific with vertebrate. Network crests are not present in vertebrate, and they have genes that are not. So these genes may be present in Amphioxus, but they are not expressed in network crests because there are no network crests at all, or limbs. So yes, I think that there are relationships, and these relationships at the end turns back to a large extent to the regulatory information associated with these genes, which is the one that is driving the whole process of evolution.
Yeah. Thank you. There's another question about how mobile elements, mobilization can contribute to these new enhancers in vertebrates.
Oh, definitely, they play essential role. It has been shown, for example, for an old paper of CTCF in the mouse lineage. There was one particular mobile element that contains CTCF sites that have generated multiple new CTCF sites in the mouse lineage, and is helping or altering the 3D architecture of the chromatin in mouse. And the same happened for enhancer. Sorry. and in fact, I think that is one of the major source of new regulatory elements, because many of these mobile elements do have primer or pre-enhancers there that can be easily incorporated in a regulatory landscape.
Yeah, I fully agree. Yeah. So another question. Some of these genes that has an increase in rewiring are related to regeneration. Have they been tested in models like ambystoma?
No, no means that we have no idea. I have no idea. We have not going farther this. Regeneration is something that need to be studied. I mean, now there are people that studying the regeneration in a similar way we are studying evolution and development,
yeah.
and I think that we will at some point reach similar conclusion, but we haven't looked at that.
This is a very particular question. Did the new vertebrate enhancers, those gained after duplication, exist as enhancers that were adopted by new genes after duplication, or they form from non-coding sequences?
So this is something we cannot do. The problem is that we cannot trace this enhancer, the sequence level. There are no conservation outside in the non-coding DNA outside vertebrates. So if you cross the lineage into vertebrate, you don't see, well, you see 10 enhancers, 10, 12. The only one that you can trace at the sequence level. So the only way to be able to trace an enhancer is either by knowing the factors that are bound, or in the way we are doing, by looking at the response to the signaling pathway. So we know that for example, genes involved in retinoic acid signaling pathway, retinoic acid receptor, okay, contains retinoic acid receptor response attack peaks. If you go to vertebrae, you find the same. The sequence may not be is not the same. The position of the enhancer may not be the same, but both genes are responding to the same pathway, and the enhancer is in a syntenic position. Is this the same enhancer? How can we say if we have no sequence? So the only way to really know it is know the total and complete language behind this enhancer. And I can tell you, because now we are doing now a lot of CSIC of multiple transcription factors. It's impressive the number of transcription factors that bound to the individual enhancers. So the language behind each enhancer is extremely complex, and it will be extremely variable between species, both in the vertebral lineage and outside the vertebral lineage. So it will never be easy to see this enhancer was there before. You can try it, and I'm sure that we have tried. You can take this enhancer that responds to signaling path to a retinoic acid in zebrafish, and it drives expression in a retinoic acid territory. But because it's responds to retinoic acid. But it doesn't mean that the enhancer is the same that the one in vertebrate. We cannot say that.
Another question is the gain in this context specialization enhancer promoter, pure enhancer promoter, or there are promoter interaction, these E-promoters?
Both. Everything.
Both.
Enhancer, promoter, everything.
And the percentage of this?
Well, something that we have not measured, but we have now another work also concentrate on CTCF itself, because we have generated CTCF mutant in zebrafish, and it's amazing how the 3D architecture needs to be maintained in order to the enhancer to be fully active. If you dismantle the 3D architecture, many enhancers that have nothing to do with CTCF itself become less accessible. So you really need to generate these confined chromatin interactions to facilitate enhancer, promoter, enhancer contact. These regulatory factories need to be there. If you disperse this because you destroy the 3D architecture, now everything is less function, less optimal, and the impact in gene expression is major, and the embryo dies. So really enhancer promoter contact is extremely important to facilitate synergy between different enhancers, because it's so complex regulation of this developmental genes and many other genes as well, that you need synergy between different elements, and this synergy can only be maintained with the right 3D architecture. If not, this is gone. So looking at enhancer individual is interesting, but it's the collective action for this finally making the real pattern and the real control of gene expression.
Yeah. Just a couple more questions, because we don't want to push you a lot. So one other question, it says, when you have gained this by duplication, these new enhancers, what happened to the old enhancers? They lose transcription factor binding sites?
But again, I don't know which are old, which are new. We cannot trace them. So we only can say that gene A respond in amphioxus to signaling to retinoic acid, but the same one of the copy of this gene, one, not all, one of the copy of this gene as a duplication in vertebrate now respond to retinoic acid and will signaling and maybe a nodal. And we have looked only at 4 signaling pathways, so there are more signaling pathways. So we cannot trace enhancers.
Yeah.
We can trace only respond to the treatment at the RNA expression level and at the time, but we cannot really compare enhancers.
Another technical question, finally, is
just one clarification.
Go ahead.
I do believe that enhancer never disappears, so the enhancer are adapted. So probably some of the vertebrate enhancers that are currently active are derived from these ancestral enhancers that have been modified. Some way they may be doing the same. The language maybe have changed a little bit, but enhancers are very valuable element and likely reused again and again, and modified by mobile element, by other issues, and by methylation, for example. So there, you don't destroy something that it is useful.
Yeah.
And plastic.
Yeah.
Plastic.
And yeah. No, I think it's very difficult to talk about the old enhancer, old and new. That's clear. Yeah, and I think we'll finish with the last question here, but it's for the PhD students. I think it's a very general question. What has been the most challenging aspect of the project, the lab work or the bioinformatic work?
The bioinformatic work has been. I mean, the bioinformatic work, especially the first part with the amphioxus, without Manu Irimia, which is a guy that you have there in CRG, and is a genius. Without him, it would be impossible. So I am, you see that the talk is very much bioinformatics, but I am total ignorant in bioinformatics. I can even work with Excel files. So that I only look at genome browsers, and I only interpret genome browsers. So I really think that the integration of so complex data has been a major challenge, especially because of this duplication event, because you cannot really assign which gene is which gene. Yeah. You have to consider families instead of genes, so it's very complex in general, the bioinformatics analysis.
I agree that the bioinformatics part is the more complex. Just a curiosity, Manuel Irimia spent a summer in my lab in Madrid, so I know Manuel very well.
I know that from the... He started with you.
Great scientist. So I would like to finish here, to conclude. I would like to thank José Luis for this wonderful talk, all the followers and people. That has been a great success. My apologies for phase 0 of the talk, but I think it has been a great success, and I would like to thank really the speaker and José Luis for this wonderful lecture with a lot of food for thought. Okay.
I really thank you for the invitation, and I think all the people that have joined the seminar. I hope that I can be, because I was not looking myself. I have problem with the camera.
Yes, don't worry. I have here more questions that I can read. Don't worry. so I'm from everywhere.
Whoever wants to ask me questions, you can send me emails. I will be happy to answer your question by email too. So,
yes.
Thank you very much.
Yes, and the other thing, we'll deposit this talk online so everybody can later check if they miss something of the lecture.
Perfect. That will be wonderful. Give me the link so I can fill it out.
We will. Thank you very much. Again,
thank you, Manel.